Okay, thank you for the introduction and for giving me the opportunity to present my PhD work. As I've been already introduced, I am a PhD student at ILL, and this is a project in collaboration with Keele University. Prohibitin is a highly conserved protein, mostly present in the mitochondria, but is also present at the cell membrane and at the nuclear level. Prohibitin in the cell membrane can have a role of protein and lipid scaffold, being responsible of some type of viral infection. At the nuclear level can act as tumor suppressor, being responsible of some type of cancer. And in the mitochondria is responsible of maintaining the mitochondrial homeostasis, that means to maintain the mitochondrial morphology and physiology. Also is co-responsible of regulate the degradation of the membrane protein of the mitochondria. Alteration or deletion of prohibitin in the mitochondria can lead to mitochondrial dysfunction, leading to pathology such as a neurodegenerative disease, as Parkinson and Alzheimer. Also can lead to a higher production of ROS, reactive oxygen species, leading to pathology, to cellular senescence and premature cellular aging. I want one moment to focus on what is the structure of the prohibitin complex, but I have to underline that most of what I'm going to say are hypotheses, because not a lot of the molecular arrangement of the complex is known. So what is known is that it's composed of these of two subunits, prohibitin 1 and prohibitin 2, and they are placed in alternative way to make a ring. And this ring is placed at the inner of mitochondrial membrane, and each of these subunits has an N-terminal helices that go to anchor the membrane. What I want to understand in my project is understanding how exactly the N-terminal helices of the prohibitin are anchoring the membrane. And the answer to this question, other than have a better understanding of the molecular arrangement of the complex, will help us understanding other question marks that there are at the moment. One of these is if the N-terminal helices of the prohibitin interact with the M-AAA proteases. As you can see here, this is a complex that is placed inside the ring. And in this way, we can understand if these helices have a role in the degradation mechanisms. Also, the recent hypotheses have been seen that prohibitin complex have an interaction with cardiolipin. In particular, as you can see from this picture, inside the prohibitin complex ring, hypotheses say that there are enrichment of cardiolipin and PE. So we want to verify if cardiolipin in particular has a role in the terminal helices insertion. Just looking a moment on the structure of the prohibitin complex, the prohibitin complex is made of an hydrophobic stretch that is the N-terminal part, a prohibitin domain, and a coil-coil domain. And what I am focusing in is the hydrophobic stretch, so the N-terminal helices. And prohibitin 1, the N-terminal helices, has an hydrophobic character with around 20% of positive charge. Prohibitin 2 has an enrichment of this charge, and this gives an amphipathic character to the helices. And so we have three hypotheses of how this interaction can happen. The first one is if the peptide behave like an integral protein, so crossing completely the membrane. The other one is if it can behave like a peripheral protein. And also we would like to answer the question if the peptide has, the two peptides has a synergism in the moment that they are together. So here I want just briefly to explain how I have chosen my membrane model. So this is how is the composition of a mammalian system cell, and you see the difference between the mitochondria and the plasma membrane. In the mitochondria, the most present lipid are PC, PE, and cardiolipin. But I want to focus your attention in the difference of percent of cardiolipin in the mitochondria and the plasma membrane. As you can see, in the mitochondria is present in 13% and in the plasma membrane 1%. And here is just a reminder of the peculiar shape of the cardiolipin. So two head group with two tail. And here I am just presenting what are the methodology that I'm going to use. Before I have used some interface technique. I start my analysis with some quartz crystal microbalance with dissipation monitoring. And then I proceed with some neutral reflectometry study. But then we want to verify if what was happening in the interface was reproducible in the bulk. So I performed some SANS experiment. So these are the QSMD experiment. I performed QSMD experiment with both peptides, so the terminal helices of prohibitin 1 and 2. And I tested this experiment, these peptides with and without cardiolipin and PE. If you look at the first table, here I'm representing the terminal helices of prohibitin 1 in presence of cardiolipin. And you can see as after that I have added peptide, I have an increase of mass of 10%. And this is due to an insertion of the peptide in the bilayer. I have performed the same experiment without cardiolipin, and we have any change of the system, so no insertion. With the prohibitin 2, the terminal helices, I performed experiment as well with and without cardiolipin and PE. And as you can see, we have a decrease of mass of 10%. And after the addition of the peptide. So we think that is due to the peptide that is able to remove lipid from the bilayer. So then I performed some neutron reflectometry experiment, and the system that I have used here is a deuterated phosphatidylcholine extracted from Pichia pastoris. This is a natural lipid that is been given to us by the research team of Giovanna Fragneto at ILL, with the addiction of the terminal helices of prohibitin 2. And here you can see the contrast that I have used in absence and in presence of peptide. And if you look at the reflectivity data, you can see as there is a changing in the reflectivity curve profile, and there is a reduction of the coverage of the bilayer. So this is due to the action of the peptide to remove lipid from the bilayer. And we suppose that it can be some hole formation. And now I have performed some SANS experiment, as I said, in order to verify if what we saw in the interface was reproducible in the bulk. So the system that I'm showing is the OPC, the OPE with cardiolipin, and this is the curve scattering profile that I have with the lipid alone. And then I have added peptide. So this is in presence of prohibitin 1, and you can see as at low Q we lose this flat plateau, and this shoulder over here at mid-high Q is a typical shoulder that we see every time that we perform experiment with cardiolipin. And in presence of prohibitin 1, this shoulder is less pronounced. And the analysis of this system is ongoing, but what our hypothesis is that we go from a bilamellar vesicle to one unilamellar vesicle elongated. And now if you look at the prohibitin 2, you can see as we still have the plateau at low Q, and the shoulder at mid-high Q is still the same. So there are no effects in presence of cardiolipin of the prohibitin 2. Now, if you look at this system, this is our lipid in presence of both peptide. And you can see as at the level of, we can say that for sure there is no synergism, and the effect that has at low Q is much less than Prohibitin 1, and also the shoulder is quite evident yet.
2 minutes, please.
Okay, thank you. So now I will go to explain what are our preliminary conclusion. So we can conclude that the membrane lipid composition play an important role in the interaction of the Prohibitin 1 and 2 with the membrane. I performed QSMD experiment and SANS experiment with several PC and P, so changing in chain length and saturation state. Also, we have our first hypothesis are that the Prohibitin 1 can face the inside of the ring, and the inside of the ring, as I mentioned before, there are hypotheses shown that is enriched of cardiolipin and P, as well is present inside of the ring the IMS region of the M-AAA proteases, and all of this can be explained with the packing parameter of cardiolipin, and the Prohibitin 2 is facing the outside of the ring. My future steps are to evaluate with CD the possible helicity of both peptide, and then I will perform some neutral reflectometry experiment with Prohibitin 1 and with the mixture of them. And I will verify the whole formation in presence of Prohibitin 2 with some AFM experiment. And I will perform some X-ray reflectometry experiment using some as complementary technique in order to elucidate if there is a rule of the concentration of the peptide in this interaction. And I want to, thanks to my supervisor, Sylvain Prévost, Anya Winter, and Trevor Forsyth, and people have helped me in performing the experiment and in training with the different technique data analysis, and the lipid lab for giving me the natural lipid. And you for your attention.
Oh,